Welcome everyone to the fourth installment of the Wild World of Bees. I'm Lincoln Best, the taxonomist for the Oregon Bee Atlas, and I'll be your host. The Wild World of Bees is a stay-at-home lecture series for you to learn more about the incredible diversity and lives of wild bees. Through this series, we'll hear from great bee researchers and advocates from around North America, and often with a focus closer to us here in the Pacific Northwest and Oregon. This series is brought to you by the Oregon Bee Atlas, an initiative to document the bee fauna of Oregon. The Oregon Bee Atlas is the biodiversity-focused arm of the Oregon Bee Project, a partnership between OSU Extension, the Oregon Department of Forestry, and Oregon Department of Agriculture. The series is also sponsored by the Mount Pisgah Arboretum in Eugene, Oregon, a great place to observe flowering plants, and the Arboretum's local bee fauna. Now, in last month's episode, I had the pleasure of introducing Cass Urban Mead from Cornell University, who spoke with us about the importance of flowering trees for bees in complex landscapes. And for those of you who missed it, in previous episodes of Wild World of Bees, they can be found on our website, OregonBeeAtlas.org, under the Wild World of Bees link right here. Or you can find her talk posted on the Oregon Bee Atlas YouTube channel, and you can search for the channel or for her talk directly. And of course, don't forget to subscribe, share, hit that notification button, and give us a thumbs up. Now, I'm very excited to announce we'll be joined by Olivia Messenger-Carroll next month. She's the author of our favorite guide to North American bee diversity, The Bees in Your Backyard. And she'll be joining us on July 9th at 7 p.m. Pacific time, and I hope you will too. Now, this evening, it's my pleasure to introduce my colleague, Professor Jim Rivers, who's the PI for the Forest Animal Ecology Lab in the Department of Forest Engineering, Resources, and Management here at Oregon State University in the College of Forestry. Jim is an animal ecologist whose research on pollinators examines the effects of natural and anthropogenic disturbances on population and community health. Now, if you haven't had the opportunity, check out his podcast with Andony Melithopoulos. It's episode number 21. Now, within this Zoom webinar, you're invited to chat with your peers using the chat function. And for those of you with questions for our speaker, please submit them using the Q&A function. You'll see those at the bottom of your screen. I'll moderate the Q&A session after Jim's talk. And so with that, Jim, I turn it over to you.
Thank you, Link. And how's that? Can you see my screen?
Not yet.
Not yet. Okay, let's try this then.
It's up. It's not in slideshow mode.
How's that?
Got it.
Okay. Well, thank you for that nice introduction, Link, and thanks for everyone for tuning in tonight to the Wild World of Bees seminar series. We are, I think we're 4 episodes in, and it's been a real pleasure to be able to be here tonight and to share with you some of the work that we've been doing in our lab. And as Link said, actually, let me check in with you, Link. I'm seeing my video. Is that, do you want me to be seen by everyone, or should I hide my video, or?
I think the recording just picks up your slideshow. And okay. So the little head box on the side there should just be visible during the presentation. And it's your choice whether you want to be exposed or not.
Okay. I'll talk to the camera just in case.
Okay.
Thanks for that clarification. So again, thanks for the opportunity, Link and Andony, to speak with you tonight about pollinator research here in managed conifer forests. And as Link said, I'm a professor in the College of Forestry, and I'm a wildlife biologist by training. And a lot of the work that I do is conducted to understand how animals interact with their environment within forested systems, particularly in areas where we have multiple use, particularly timber production here in Oregon. And I think that this group, probably more than any that I talk to, really understands and appreciates how important pollinators are. And really understands the weight that they have in terms of influencing our global food production, our food security, as well as understanding how our natural ecosystems are influenced. So much of the agricultural crops that we have benefit in some way from pollinators. And we also know that in a wide variety of ecosystems, we have pollinators that are benefiting a lot of our wild plant species. And in fact, when we talk about pollinators, we typically are thinking about bees in agricultural settings or other open habitats like meadows and grasslands. But what I want to focus on tonight is managed forests. And specifically, what I want to ask the question and answer it for you is, do managed forests provide habitat for pollinators? I am, as I said, I'm at Oregon State, and a lot of the work that we do is in the Oregon Coast Range. And this is an area that is really central to conifer production. It's a global leader in producing softwood products. And the photo that you see in front of you is what might be considered a typical landscape level photo of the Coast Range. And what you can see is that there is a lot of forest, there's a lot of trees, but there's also a fair bit of variation in terms of the age class of trees. So in the foreground, there's a recently harvested stand that's been planted back to Douglas fir, which is one of our key planted species in the region. It's also a native species. And we have older stands as well in the background. And so most people haven't really been thinking about this topic when they look at this landscape to ask the question, do we have pollinators in this system? And in particular, how are those pollinators influenced by the management activities that undergo in this location? So what I want to do tonight then is center my talk around three different questions. The first of which is to discuss the key knowledge gaps for pollinators in managed conifer forests. And really the starting point here is to ask the question, why would we expect to have pollinators in these areas? And then what does the science say about what's out there? The second question is focused on one of the empirical studies that we've undertaken in my research group, and that is to understand how a particular type of forest management treatment, biofuel removal, may be influential on bee communities. And I think you may be surprised by how bees are influenced by this particular management activity. I certainly was when I conducted this work. And it really got me thinking about how important forest management effects on bees are. And then the last part of the presentation tonight will focus on some work that we are just starting to wrap up. We are asking the question, how do pollinator communities change with time in young, regenerating, intensively managed conifer forests? And when I say pollinator tonight, I often am thinking about bees. That's my favorite pollinator group. But the thought is that oftentimes when we talk about what is good habitat for bees, we might also be talking about what's good habitat for other groups as well. So in terms of this first question, in terms of key knowledge gap for pollinators, if you spend any time in the Oregon Coast Range, you might come across an area such as you see in this photo. This is a clear cut that is on Weyerhaeuser property. It is about 3 or 4 years after harvest. And what you will notice is that this is an area that has been replanted back to Douglas fir. We see a number of Douglas fir trees in the foreground here, as well as across the stand. And at first glance, this doesn't really look like it would offer much in the way of pollinator habitat. But if you've got closer to the ground, you kind of adopt a bee's eye perspective, what you'd find is that there are a surprising number of floral resources in this particular stand. And that's true, in fact, of many stands, even those that are intensively managed and have been subjected to herbicide. We live in a temperate rainforest, or at least the Coast Range is in a temperate rainforest, and that means that a lot of the flowers that we have a great opportunity to grow, and grow they do. We really have incredible productivity in these areas. And just to highlight a couple of really common plants that we find in these forests, in the center top photo is a photo of salal, and in the this yellow plant on the middle right photo is Oregon grape. And those are two common understory plants that we often walk by. We don't really think of them as plants for pollinators, but it turns out that they can be pretty good in terms of their nectar and pollen production. So from the standpoint of one of the key needs of bees and other pollinators, we do have floral resources in these areas. Of course, it depends on how we manage these stands, but we do have that as kind of a baseline. We also know that many bees are ground nesting species, and a lot of these ground nesting species require exposed soil to nest. And in fact, about 70% of the world's bees are thought to be ground nesting species. So when we manage our forests, we also have opportunities in roadside areas such as you see here, where there may be openings for bees, there may be scarification of the soil, and it turns out that ground nesting bees don't really like duff and they don't really like litter, and they won't typically burrow through that material to get to exposed soil. So this photo shows what looks to be a pretty banged up roadside area, at least from our perspective, but to something like this sweat bee, this small halictus, this might actually be a pretty good spot to try and set up a nesting site. At the same time, we have other bees with other nesting needs. Seratina is one of our small carpenter bee genera, and oftentimes you can find this particular group in many of our cane plants. This is a Himalayan blackberry that has broken off, and you can see that there's a hole here in this photo. And if you open up that cavity, what you'll find is that there is actually a nest of a small carpenter bee. So not only do we have floral resources, but we also have potential nesting substrates for bees and other pollinators within these managed forests. So it stands to reason that we might find bees there. So along with my colleagues, I asked the question, what do we know about bees and other pollinators within these managed conifer forests? As we were starting to embark on this work, we really wanted to understand what the literature was out there and what it said about pollinators within these particular areas. And I have to admit one of my deep dark secrets, and that is that I'm formally trained as a behavioral ecologist, and I study birds. And I only came to bees in the last 5 or 6 years that I become indoctrinated in the world of bees. So as part of my learning process, I also wanted to understand what did we know about pollinators in managed conifer forests. And oftentimes what we do when we're trying to, as scientists, to try and understand what the knowledge gaps are is we conduct a formal literature search. So along with some colleagues, we decided that we were going to try and understand what sort of work had been done on this topic. And we focused around pollinators within temperate managed conifer forests because that's the type of system that we work with here in Western Oregon. And so we started this search with Web of Science. This is a search engine that accumulates journal articles and peer-reviewed publications. And we took a number of relevant keywords and put them into Web of Science to see how many unique papers we could come up with. And it turns out that there were over 1,200 papers that we initially had a hit on using all of the keywords that we tried. So we actually were surprised by this, thinking that maybe there was a fair bit of research out there. Of course, keywords don't always tell the full story on a paper, so what we wanted to do was to evaluate them a little more closely. And to do that, we went through each of those papers and we read the title and or the abstract to figure out if we could exclude the paper as being one that was tied to this particular topic. And once we got there, that reduced our pool by quite a bit, down to 66 papers. And then we decided to take one more step, and that was to actually look at the paper, read through it, examine it more closely. And at the end of the day, or I should say at the end of the days, because this did take some time, we ended up finding only 14 papers that addressed studies of pollinators within temperate managed conifer forests. So just over 1% of the original series of papers that we found. And that suggested to us that there really wasn't much out there. In fact, it was less than we had thought. We figured that we would find more papers than that. We decided to go through and look at the papers and try and figure out what sorts of characteristics they shared. And what we found was that this group of papers were really limited in terms of their geographic scope. Only 4 countries were in this pool of papers. The majority of them were focused on bees, which makes sense given that bees are often the most abundant and important pollinator within forested systems. And most of these studies were conducted in pine and spruce forests. We also took one more step and asked about the different types of management activities that may have been addressed in these studies. And we came across these 4, what one could consider management activities, or at least things that are under the direction of managers. And most of those studies were focused on looking at different aspects of timber harvest, clear cuts versus shelter woods versus gap selection. There were some papers looking at prescribed fire and vegetation control, and then a handful of papers that were looking at how communities change relative to the age of stands. So what we wanted to do with this information is to summarize it. And as we talked more about it, what we realized was that we had an opportunity to outline some key research themes for folks who are interested in addressing this topic in more detail. And so in addition to publishing a paper that had a summary of what we found from the papers, we also put forward what we call a research agenda for pollinators and managed forests. And that agenda really has two key themes that we want to hit on in the future. The first of which is that we reason that it's really important for us to start getting baseline information about pollinator communities. We often hear about declines in populations of pollinators, but we don't have a good handle on what populations have been through time. And in forested settings, we don't even know what species are there. We really lack that information from a long-term perspective. So just getting kind of basic science information about which species are there, how they vary through time and space is really valuable. We also argued in that paper that we really need to understand not only what's there, but how what is there are influenced by different types of forest management activities. And in particular, forest management that can be conducted within an experimental context, because we have a stronger ability to consider what really is driving the pattern when we can have an experiment and we can assign causation. And when we talked about forest management activities, we're not just talking about what we would traditionally think about in terms of forestry, but really any type of land management activity in a forested setting. So something like creating new roads or spraying exotic species in roadside areas, those sorts of things also have the potential to influence pollinators. So I think this gives a good overview of why we would think that pollinators would be in managed forests in the first place, and then also sets the stage for the work that needs to be done with this particular group. I want to change gears a little bit from providing some background and some overview to actually sharing some of the results from one of the studies that we've conducted in the last several years. And this study is focused around understanding how different bee communities or different species within a community respond to biofuel treatments within these intensively managed conifer forests. And this was the first project that I was involved in as a wildlife biologist that focused in on bees. And I think I'll walk you through the steps as to how it got me thinking about some of the work that we need to do in this particular field. Now, I mentioned this a little bit earlier, but it's important to note that here in the Pacific Northwest, where we really are a global leader in conifer timber production, one of the ways in which we manage our forests is to grow trees. We plant them in these plantations, and they grow very quickly. And then there's a clear-cut harvest. And what you're looking at here is one such clear-cut. It is an area that is harvested all at the same time, and then shortly thereafter, it's planted back to a particular species. Typically, it's a commercial species like Douglas fir, but that's actually a statute here in Oregon. We have to plant back within a certain number of years, and not only that, but the trees have to be in what are called a free-to-grow situation. And that's important because it helps to make sure that when we harvest trees, we're replacing them with additional individuals. Now, when we harvest in this manner, we often are trying to maximize the growth of these planted trees. And so as part of the post-harvest management, we usually go through and take up all of what are known as logging or more technically forest harvest residuals, and we mound them up. And we do that so it doesn't impede the growth of the trees that are planted. And in fact, if you can see my cursor, in this particular clear cut, there's a mound of logging here, there's another one here, another one here. Well, years ago when gas was going up over $5 per gallon and there was a lot of concern about dependence on foreign oil, the question came up as to whether or not these logging piles could be used to produce biofuel, in particular jet biofuel. Usually managers burn these piles so they're not used in any other way, and they're a bit of an impediment. So the question became, can we actually use them to create a green biofuel? And so the Northwest Advanced Renewables Alliance was a project that was instituted. It was a large-scale project with a number of different universities. And the stated goal was to create jet biofuel. And it seems a little futuristic. It seems a little crazy to think about. But the idea here is that instead of burning those slash piles, what one would do is to chip all of that material on site, put it into a truck, and then ship it off site, and then have a series of chemists and engineers and other individuals who would go through and ultimately get those materials into jet biofuel. And much to many people's surprise, this worked. There was a cross-country flight from Alaska Air that flew on sustainable biofuel that was taken from some of the NARA sites, including, I believe, the Oregon site, which was one of several across the country. So our group got involved with this project not because of our expertise in chemistry or in engineering, but because of our expertise in wildlife. And in particular, what we wanted to understand was what were the environmental consequences of this particular management action. So our study really had two very closely related objectives, the first of which was just to go out and describe what the bee community was within this large-scale clear-cut, and I'll give you a sense of what that location looks like in just a minute. But given that we don't have information from the Pacific Northwest, that was really kind of a very easy objective. Go out there, just determine what's there, kind of basic science. The other part to that, though, was to focus on the management piece and understand how this particular form of forest management might influence local bee communities. And as part of the formal experiment, there was a biomass removal treatment and there were soil compaction treatments that were applied together. And I'm not going to spend time talking about the soil compaction treatments. The short story there is that they don't really influence bees in any way. It's really the biomass removal, so I'll spend time talking about that. But you can imagine that as you are thinking about bee communities and thinking back to a few slides ago where we were talking about some bees nest in the ground and some are stem nesters, you might expect that certain groups might benefit more than others with respect to this particular type of management. So we conducted this work at the NARA site that was located near Springfield, Oregon. This is on the west slope of the Cascades. And this was a location, this is a large clear-cut harvest that occurred, pretty level area. There were very strict considerations for where this site could be located, given the heavy equipment that needed to be used in this experiment. In 2013, in the wintertime, this was a 70-year-old stand. It was harvested in spring, and that's when all of the treatments were implemented. The following spring, it was planted back to Douglas fir, and then that occurred on 28 different plots. And this photo is taken from Google Earth, and you can even see the treatment differences here. For example, this is one of the removal plots right next to one of the less intensive plots where biofuels were not removed to the same extent. So it's important to note here then that this was a 70-year-old forest, and it was a forest that, as best we can tell, would not be a good forest for bees. So essentially, we're starting from scratch in terms of the bee communities. And our sampling occurred, as you'll see in a moment, in 2014 and 15. So this really represents the first full season that bees were on the site. And I mentioned there were two different treatment types. The biomass removal type consisted of 3 different treatments. The first 2 treatments, which are labeled A and B here, are where the bole of the tree was removed and everything else was left on the particular plot. That's typical of what we do with harvest nowadays. The bole, you can think of it as the merchantable timber that can be extracted as a log. There were also 2 treatments, C and D, where the entire tree was removed off of the plot. So not only the logs, but all of the branches as well and the tops. And then there was an additional treatment where the entire tree was removed and then the forest floor was removed as well. And this includes removing all of the litter, all of the duff. And as we move from treatment A to treatment E, we increase the intensity of the treatment and the disturbance as well. As I mentioned, the soil compaction treatments are not really critical to this study, except to say that we had to have a consideration of the soil compaction for some of the plant work that was going on. So you probably wonder what this looks like on the ground. Well, this is one of the, this is the lightest treatment. This is where the trees were removed. You can see there's a lot of branches that are left behind, a lot of logging slash here, and there's no soil compaction, no heavy equipment was used on the plot proper. Everything was, the trees were felled off of the plot and then removed. On the other end of that disturbance spectrum, this is what it looks like. And if it's a little shocking to you, it was certainly shocking to us when we got out there and started looking. This would be a regular forest, except it's had all of that litter and all that stuff removed. And you can see the Douglas fir that had been planted back onto the site. Now, once we got out there, we started thinking about the bees that might be in this area. We really started to question our sanity a little bit, because if you look at this site and think that there might not be much in the way of bees, that's exactly what we were thinking. In fact, we were quite worried about that when we first started sampling. To sample these sites, we ended up using blue vein traps, and a lot of you are familiar with blue vein traps. They are traps that are reflective in the UV spectrum, and they are attractive to bees because bees perceive them as flowers, and we can be putting these out for a day or two at a time, and they are a pretty efficient way to sample. So in each of our 28 plots, we established 3 of these blue vein traps. You can see there's 2 here in this particular plot while we were sampling. We had 3 different rounds that we sampled throughout the year, and then we sampled across 2014 and 2015, and that gave us just over 1,000 trap days. So we started this project holding our breath a little bit, wondering what we were going to be able to accomplish, and really a little concerned about whether or not we'd be able to find bees in this site. And I should point out as well that this site was also subjected to herbicide after the Douglas fir were planted. So the experiment was to exclude any sort of other vegetation outside of the planted trees. So it really paints a picture of a pretty bleak landscape. That said, you can imagine our surprise when we started to trap bees, and as we moved through time, we ended up catching a large number of bees, over 7,500 individuals over the course of the 2 years. And those individuals represented a wide diversity of species, 92 species or morphospecies in total, representing 21 different genera. And suffice to say, it was quite a surprise that we had this much diversity, because if you talk to Link and you talk to Andoni and others, they'll tell you that we don't know how many bees we have here in Oregon, but there may be 600 or 700 or 800. And if those numbers are accurate, what we found here in this one site was nearly 1/6 to 1/8 of the bees here in Oregon. So it was really, really quite a surprise to us. Not only was it a surprise how many bees, but also how the bee communities changed through time. The first year we caught upwards of 2,000 bees, but that quickly went up in the second year by about more than 3 times. So what we're seeing here is the population of bees in general, not any one individual species, getting bigger as we move through time. And if you're wondering about species, we also saw that about 20% more species were detected in the second year of the study. And if you think back to that photo that showed the aerial view, and I was discussing how that stand used to be a 70-year-old stand of trees that likely didn't have bees. What we're probably seeing here is the development of these bee communities. We have a large-scale disturbance that opens up the canopy, and we start to see bees populate that area, which is, I think, what we're seeing here. And it would have been great to continue this through time, but unfortunately, this was only a 2-year project. When we looked at which species or which life history traits we captured, what we found was that we had mostly ground nesting bees in our particular sample. So upwards of, I think it was 91% or perhaps a little bit more, were ground nesting species. And these are the different genera that we captured. So a lot of you will recognize Halictus and Lasioglossum and Agapostemon as ground nesting sweat bees. We did have a few other species that we detected that were either cavity nesting bees, shown here in green, or twig nesting bees in blue, a few Ceratina, but by and large, most bees were ground nesting. We also wanted to look at those different treatments to see if we could detect differences in the number of species across the 5 different treatments that we had. And so what we're looking at in this picture are just the average estimates for the number of bees on each of those 5 different treatments, starting with the lowest intensity treatment in the form of A, going all the way up to E. And what we found was in the first year, we had significantly more species in this treatment E. This is where we had the duff and the litter removal. And we found the same thing in the second year. Both years we had averages, and if you take that over the entire project, what we find is that there really is a treatment difference, but it's driven by this treatment. So from a statistical standpoint, these are all very similar within a year, but these bees in these treatments are, in treatment E, are different relative to the other ones. This figure looks at species richness. If we looked at abundance and just tallied up the number of individuals, we'd see a very similar pattern. So what we're finding is that this more intensely managed treatment actually had more species of bees and more individuals. Well, another way that we can look at this is by looking at ground nesting bees. And what we wanted to do was use these traps. These are called soil emergence traps. They look a lot like a tent, but they don't have any bottom to them. So we established these emergence traps at both ends of the treatment spectrum, in the least intensively managed stands, and then some of the plots that were most intensively managed. We put them out for over 3 months, and we wanted to see what sort of bees would ultimately come out of the ground. And we were a little bit dismayed because we had them out for over 3 months, and we ended up catching 15 individual bees of the species that you see here. And in fact, we only did this in the second year, or I'm sorry, only in the first year, because there wasn't that much in the way of results. But of note, all of the bees that we captured were in that very intensively disturbed treatment. And if you think about it, we captured 15 bees, and those small traps don't sample a very big area. In fact, the traps that we had covered, on average, only about 4.32 meters squared. So across all of those plots, we were sampling a very small area. And it turns out that those plots, if you calculate how big of an area they cover, it's more than 24,000 square meters. So we can take these numbers and multiply them together and come up with an apparent abundance estimate of taking 3.47 bees per meter squared times 24,000 plus meters squared. That leads to over 84,000 bees. Now, don't think that I'm arguing that there are 84,000 ground nesting bees out there, but what I am trying to point out here is that there's probably lots more bees there than we might anticipate, particularly when we sample a small area. And just as an index of that, when we conducted our field work in 2014, in just 6 days of trapping across that field season, we caught more than 1,600 bees. So that's a lot of bees in just not even a full week's day of trapping out there. So I think a lot of these sites are attractive to bees, not because they have floral resources. It turns out that almost half of our floral resource surveys turned up 0 flowering plants. It turns out the managers did a good job spraying herbicides. But instead, what we think is going on is that these stands were being used for nesting and not for foraging. We based that on a couple of different lines of reasoning, one of which is that we had observations of bees that were mating on site, and we also had a few nests that we found. And that, coupled with the great majority of our bees being classified as ground nesters, really suggests that bees weren't coming to these sites for food, they were coming to breed. And in fact, at the periphery of our plots, in other locations close to where this study was conducted, there were floral resources. So what we're hypothesizing is that these bees were moving over into those other areas to get the nectar and the pollen that they need to maintain their populations. So the key finding from this study is that we had a surprising amount of bee diversity within an intensively managed conifer forest. And what's really striking to me is that this is one large clear cut in the west slope of the Cascades. We didn't have 15 or 20 or 30 plots. We had one location with nearly 100 species of bees. And I think the take-home point here for managers is that activities can influence bee habitat even when there's no thought about bees in the first place. And so I think that really resonates with me in thinking about all of the different ways in which how we manage our forests can ultimately be influential on the bees that are there. So the second study that I wanted to share with you tonight is focused on understanding how these communities change through time. And as you saw with that, with the NARA study looking at biofuel, we saw this very rapid change in these communities. Once trees were removed and the canopy was open, bees seemed to really respond and move into those areas, and they started to build through time. And so we got really interested in this question following on the heels of the biofuel study. And what we wanted to know is how do these communities change during the regeneration stage of forests? So if you don't spend much time out in the Coast Range, or you don't know what a clear cut looks like, this is a clear cut in early spring. You can see that it's been planted. There's just a couple of Douglas fir here and there, but by and large, this is not a site that has a lot going on for it. But very quickly after harvest occurs, because we live in such a productive environment, just a couple years later, we start seeing changes. And as you can see here, we have, in this particular location, we have a lot of foxglove that's starting to bloom. We have roadsides in those areas, and we really think that pollinator habitat starts to improve shortly after harvest. We also know that managers have the goal of closing the canopy of these managed Douglas fir forests as quickly as possible. And in fact, in some cases, it can be less than 15 years before a view like this in the upper right starts to look like what you see in the lower left. This is a Douglas fir plantation. It is managed in very much the same way as the top two photos. It's gotten to the point, though, where all of those trees are starting to, the canopy's closing, and they're starting to exclude all kinds of plants from the understory. And if you know anything about bees and you look at this photo in the bottom, you'd say, boy, that doesn't look like a good place for bees. So our hypothesis was that the window for pollinators would be pretty short in these intensively managed conifer forests. So we were able to go out and get some funding from the USDA NIFA program. And we've been able to work with industry and partners and with the Oregon Department of Forestry to survey pollinators on land that is being used under operational conditions. So we haven't asked them to do anything different than they normally would do. We've just asked for permission to sample on these sites because we want to understand what's there under contemporary conditions and to get that background information before we start asking questions about how we might be able to tweak the system one way or another to improve it for pollinators. So we started this project in 2018, and we selected 60 sites that were in the Oregon Coast Range. Most of them are outside of Corvallis because that's where our base of operations is during the summer. And those sites were 0 to 35 years post-harvest, which means that some of them had been harvested in the wintertime, and we went out and sampled them in the spring. Others were a little bit older, and then there was a handful of stands that were anywhere from 20 to 35 years old, because we wanted to understand what happens in those older, more mature forests. Unlike the biofuel study, instead of looking just at blue vein traps, we also put out pan traps. This is one type of pan trap that we used. We also had some that were blue in color and yellow and white. And we also used targeted netting off of flowers. That gives us a lot of good information, not only about what's there for pollinators and flower visitors, but also what plants are on site as well. Now, we conducted this over 2 years, but because we've had such a huge number of specimens, it's taken us a while to get a lot of those identified. In fact, we only recently got back our 2019 samples from Link. So what I'm going to do is share with you results from 2018, but as best we can tell, they're representative of both years of our study. So in 2018, what we found, again, was that there was a huge number of specimens. Over 12,000 specimens were collected as part of this project. And we found that native bees were really the dominant pollinator group that we captured in the course of fieldwork. So this figure shows here how much of our collection in 2018 in young stands, that is to say stands that were 0 to 4 years post-harvest, were bees. And it works out to be almost 90%. We do have flies, we do have butterflies, and we do have beetles as well, but for the most part, it's mostly bees. And it continues to be so through time, although you'll note that the number of flies start to increase as we get into older sites. We can take this information and look at it on an individual stand level, and we can do that by both our passive trapping, that is to say our blue vein traps and our pan traps, and then also look at it through netting. And usually what we find from other studies is that we get a lot more specimens when we use our passive traps because they're out there for a much longer period. So if we are, so this first figure I'll show you is representative data collected around our pan traps and our blue vein traps. And what you're looking at here are the number of individual specimens that we collected on each stand on the y-axis, and then the stand age is on the x-axis. And what you'll note are a couple of things. The first of which is that there's a lot of variation, even within a given stand age. Some of these young stands have huge variation in terms of the number of individual specimens collected. And I should point out here that we're collecting across 3 sampling periods that represent much of the flight season for bees. So a lot of variation. You'll also notice that, again, bees are the most abundant group. They're represented most by these green dots. And then beetles follow them with the red dots here. The other thing that you'll notice is that there's a real strong drop-off right around year 12, 13, and 14. With our passive traps, we just don't seem to get much in the way of any pollinators or flower visitors in these stands. So this is, again, a lot of variation, but early on we see kind of a peak and then it starts to decline with time. So keep in mind the scale here, we're looking at 0 to 1200 on the y-axis. We'll move over to netting off of flowers and you'll see that we move from 0 to 40. So quite a few fewer specimens when we are out netting off of flowers. And we knew that going into that, but as I said, this gives us a lot more information. You'll also notice that it gives us a different result in the sense that instead of beetles being the second most common group, we actually see that flies are. So there's a lot of syrphid flies that we find. There's some bombylids, the bee flies as well. And those are represented here by these olive green dots here. Again, bees are really the dominant group. And again, things start to really taper off by about 14 or 15 years in terms of stand age. And you will notice that there's a couple of blips here. We do get a few bees here. We do get a few flies. It's interesting to note because we thought that those sites would not produce any sort of pollinators. We didn't catch any with our passive trapping, but it turns out that there are some small microhabitats within those older stands that were close to where we were sampling. So we think this is reflective of some gaps in those older forests. And it's really prompted us to think about the value of doing some research on thinning and how thinning might be a way to enhance pollinator habitat, particularly given that the Forest Service doesn't do large-scale clear cuts at the current time. They do a lot of thinning operations, and they're interested in pollinators as well. So that's got us thinking about that. So this is a pretty strong relationship here in terms of when it stops, and it's consistent across both of these sampling types. So the question is, what's driving this relationship? Well, one thing that we think is going on is that flower density really gets down quite a bit as you get stands. They get older, they start to close their canopy, and there's less and less sunlight hitting the ground. And that's shown in this relationship here. This is stand level flower density, and you can see that there's a bit of a peak early on, but then it starts to go down almost to 0, and it bounces around a little bit here. But again, interestingly enough, one of our older sites has what appears to be a pretty good group of flowers. So that's one thing that's tied to pollinator abundance in these stands. But what we think is ultimately controlling pollinator richness and pollinator abundance is that stand level canopy cover. And if you think back to that photo I showed of that stand just about 15 years of age, you can see that canopy cover, the trees start to fill out and canopy cover becomes 100%. And that typically happens by the time by the time the stand gets to be about 15 years of age. And there isn't much in the way of variation after that. What this is telling us are really 2 things. The first of which, in these intensively managed forests, managers are doing a really good job growing trees and getting that canopy to close, because that is an important part when you are producing timber. The other thing that it tells us is that if managers are interested in conserving pollinators or providing additional habitat, they really need to think about these early years, up until maybe age 6 or 8. That's when the most bang for one's buck happens in terms of pollinators. Those are the greatest communities where we have the greatest opportunity to improve or provide habitat for this group. Now, we were going to be sampling this year. We had planned to have a third year on this project, but because of the COVID-19 situation, we've been unable to sample. So this project is going to end after 2 years of sampling. One of the real interesting things, though, and what I had hoped to continue sampling on these sites, is that we found a really strong reduction in bee abundance in 2019 relative to 2018. So what you're looking at here is the total number of bees that we captured within our all of our trapping efforts in 2018. And you can see that that was markedly reduced last year, 2019. And it wasn't just one particular group. These are all the 5 families of bees that we captured. It ranges from a third of our captures decreasing in halictids, the ground nesting sweat bees, to almost or more than 3/4 of our apids decreasing. So from one year to the next, we saw this huge range. And it's not just a decrease, but you'll notice that it wasn't a proportional decrease. The halictids went from being second to the apids to being the most dominant species. So there is a lot of change from one year to the next, which would have made this a really interesting data set to continue into the future. What was really surprising and what was really jumped out at us was the number of bumblebees that were captured in 2018 to 2019. Here we saw a decrease by 90% between the 2 years. So 11 species in 2018. We captured almost all of those species again, but there were very, very few. Only 10% of the species that we, or 10% of the individuals were encountered. And this, if you think about bumblebees and how important they are, we know that many of these are generalist pollinators, and they're not only important in natural systems, they're also important for a lot of the agricultural crops that we have, like red clover and blueberries, cranberries. So what this effect has, we don't really know. We just know that there's a lot of variation from one year to the next. If you're watching this, you're probably wondering, well, what drove that variation? How could you have so much of a decline from one year to the next? And if you were in Western Oregon last year, one of the things that you probably appreciated was that we had cold and wet periods throughout much of the growing season. What you're looking at here are mean daytime temperatures across the entire season of when we were sampling. So day of year 100 is in April, and this goes out into August. And you can see that stands that are younger are those in dark red. Stands that are older are in blue and purple. And there was a real demarcation in that the younger stands got a lot warmer over the course of time in 2018, but that wasn't the case in 2019. This spread of points was much smaller, and you can see that a lot of these young stands were actually much closer to what the temperature conditions that we would have found in an older stand. And in fact, many of these events where we had warmth and then we had cold were associated with wet periods as well. So it really was a challenging time for bees. Whether or not this has long-term repercussions, we just don't know, and that's something that I think we would love to know more about. So we've talked about a lot of things tonight, and I hope that, at least for this last section, I think that you can probably see how we built on some of our knowledge, some of the knowledge gaps that we found, and been able to show that these pollinator communities are incredibly dynamic, not just from one year to the next, as you saw in that last slide, but also from the time that harvest occurs to the time that canopy closure occurs. So if we are thinking about these stands again from a management perspective, we really need to be thinking about stands that are young and shortly after harvest. That's going to be where we can get the most out of our management actions if we want to try and have management for pollinators on top of some of the other uses that we have. So if you're to leave with 3 points for tonight, these would be the 3 points that I would hope that you would take with you. The first of which is that many of us don't think about managed conifer forests when we talk about bees and other pollinators, but we really have some interesting opportunities for pollinator conservation. And I didn't talk about this, but one of the things that we would love to do is start linking bees in forests to bees in agricultural lands, and really be able to see if they may provide colonists, or there might be opportunities for populations to move between those two areas. But nonetheless, if I've got you thinking about managed conifer forests for the first time as pollinator habitat, I've already done my job. The second take-home point is that what we do and how we do it within forest management and the activities that we undertake can create habitat, even if we don't mean to do so. So that gives us, I think that gives us some, that helps us make the case that when we are planning harvest, we're planning roads, we're planning on spraying exotic plants, we really need to think about how pollinators may be impacted, because oftentimes we undertake management for timber production or for other purposes, but those activities can actually have a pretty big influence on bees, either positively or negatively, as we saw with the biofuel study. And then the last point is that we have a very short window for pollinator habitat when we talk about these intensively managed conifer stands. So the work that I've done, I presented it tonight as a collaborative endeavor. These are a number of different colleagues that have joined me on these projects. I've learned a ton from them. They help keep me in line, being a reformed ornithologist who studies bees. And I'd also like to not only acknowledge them, but also the funding groups and all of the support that we've got from a number of people over the years. And with that, I want to thank you for tuning in tonight, and I'm happy to answer any questions that we have.
Well, thanks so much, Jim. That was fantastic. I'll moderate the questions. So if anyone has any questions, please type them into the Q&A bubble at the bottom, and we'll get those in the queue. So let's see, from Doug Cooper, he asks, in the biofuel study test site, how was litter and duff removed? In treatment E, looked as if it was done mechanically, yet that must have been very costly and maybe not a viable practice.
Yes, I didn't get into the details of how those treatments were implemented, but it was done with harvest equipment, and it was mechanically done, and it would be costly. My take on why that was used is a lot of times when we conduct experiments, we want to know the full suite of the response in a given, to a given condition. So, for example, if we are looking at herbicides and their impact on birds, for example, we might have the typical herbicide levels that are used or the range, and then we also have a control where we don't do anything, and then we have an extreme level where we put on herbicides to a degree that others might not do. So I think you're right, Doug, in that that might not be a viable practice. At the same time, there was this was in an era where there was a lot of question about the cost of a barrel of oil and how high that was going to go. It was reaching record highs, and so in that equation, I think a lot of people were thinking, what could we do, and would it be viable eventually? I think what that study found was that just producing the biofuel was energetically expensive, and it was probably not something that could be done over a long term, at least within forested areas. But there were other habitats that were being assessed as well. So there was some biofuel in grassland areas too. So the forest was part of kind of a larger study, seeing whether this was viable or not. And what we don't see is people enacting that practice. So I think your point's a good one. It's not something that we're going to now, but at least we have a good handle on how it might work down the road.
Yeah, it's a great answer, Jim. Thanks. From Diane, was it possible to do a pre-test of the area? And what she means is trapping ground nesting bees before the area was clear cut, or was all of the trapping post-harvest?
So I think, I assume that this is referring to well, it could refer to either one of our studies. We did not pre-test the area. So it could be the case that there were some patches in there. As we saw in the latter part of the presentation, there are some areas that are older sites that do have gaps, and gaps happen for a number of reasons in forests, and that's an area that we don't have a good handle on in terms of the type of habitat it may provide for pollinators, and I think that's an area that we need to go. For the most part, doing a pretest in these areas, we've done these sorts of studies with birds as well, and we know that the habitat requirements of certain species just aren't met by those 70-year-old, 110, 120 foot tall trees. So I'm comfortable saying that it was highly unlikely that we would have had good habitat for bees, particularly for that many different species. And the work that we did in the Coast Range, looking at the age of harvest, would suggest that there are some bees there, but they're just nothing nearly as what we would find in some of these open habitats that are younger forests.
That's great. Let's see. From Doug again, what's the home range of bees? How far do they travel for forage?
That's a great question. It depends on a couple of things. One is whether or not it's a female that has a nest. So males and kleptoparasitic bees aren't tied to a nest. We call that central place foraging, where a bee or a bird or anything that has a nest site can only go so far before it has to return to that nest. If it's a male or a kleptoparasitic bee, they can keep going and keep going. They don't have to return to that location. What we do know about females that are nesting is that the area traveled by a bee is strongly dependent on the size of the bee. So some of our really tiny bees may only travel 100 meters, 200 meters in their lifetime, whereas a larger bee, like a bumblebee, might be traveling several kilometers across the landscape. It's not an easy question to answer because we can't put tags on bees like we can a lot of organisms and get an idea of how big of an area they use. So there's been a few studies. One was really interesting because they had specialist bees and they were testing them on the plant that the bee would go to in a field that had no other forage. And they had this, I think this was in Germany or in Switzerland. It's probably more of a Swiss thing because the Swiss always come up with these ideas. But they actually had the pots on a rail system and moved the pots to different distances and marked the bees individually to see how far they would travel. So in terms of our study, we would anticipate that some of the smaller bees would be having to get their food pretty close to the plot on which they were nesting. Finding bee nests is also really challenging as well. So there's a couple things stacked against us, but it's safe to say that it's pretty variable and that bigger bees travel a bigger area and can pollinate a bigger range of plants.
Great. From Tony, was there any difference in the number of bees caught in the traps closer to the floral resources versus traps deeper into the logged area? I'm going to guess that was for the second study that I talked about, and we don't really have much in the way of differences because when you're sampling in the area where we are, there's the potential for what we call edge effects. So roadside areas might actually be not the worst habitat for bees because they're open areas, they're warm, there's lots of flowers. There's oftentimes cut banks on the side of the road that would allow access to soil. So what we wanted to do with all of our sampling was to sample in the middle of that plot and get away from those potential effects, because when you have edge effects, you want to make sure that you get far enough away from them so that when you're sampling, you're really sampling what's representative of that plot. And so when we put out the passive traps and we put out transects to net on, they're all in the same location in the middle of the stand. And I didn't point that out, but in both of the studies, we had the sampling points right in the center of the stand, so we would be able to say something representative of that particular stand age, or in the biofuel study, the treatment combination that was on that plot. Great, thanks, Jim. Let's see, from Phil, are there plans to share this information with federal land management agencies affecting West Side and ONC lands?
Yes, a lot of the work that I do is sharing this with through presentations like this. We had the Pacific Northwest Bee Conference. I think we had quite a few folks from agencies that were there. We had an early cereal conference Early Stroh Forest Conference just about a year ago now, and I gave an overview of some of this pollinator work. So it's really getting to be of interest to a lot of state and federal agencies, and we've been able to bring those folks in for workshops and also share these results either through publications that we share with them or sometimes just getting on the phone and talking to them about the research that we're undertaking. We don't want to do this in a vacuum and be in the the ivory tower. We want it to be relevant to land managers, and so a lot of the work we do is getting the word out about what we find to a variety of different groups.
Perfect. Thanks, Jim. Now we are starting to run out of time. It's already 8 o'clock. So we'll do, I think, one more question, and then I'll conclude with some notes on upcoming presentations. Now, Ellen asks, what are other research questions that are on your list for managed forests and pollinators in particular, and ecology in general?
My list is long. If anyone's interested in the details, if you go to our lab website, which is shown here, and you go to the publications page, that Journal of Forestry article is online and you can download it. And there's a lot of information in there about different types of projects. I listed kind of the high points and the big themes, but we go into a lot more detail. I think there's a lot of really important work to be done around herbicides. I think there's a number of topics related to just general land management as well that can be tackled next. So I could go on, but in the interest of time, I'll refer to that paper as a place where we've put some of our best ideas for folks to consider.
Thanks, Jim. That's fantastic. Thanks for joining us. I know that you have all sorts of plans for studying pollinators and landscapes, and I'm pretty excited about that. So let's see here.
Thank you for the opportunity to talk, Link and Donnie. Appreciate it.
All right. So, folks, if you're interested in joining the Oregon Bee Atlas to help document the Oregon bee fauna and become a master melitologist, someone who studies wild bees, you can find information on our website, the OregonBeeAtlas.org website, and follow the Master Melitologist link, and that'll be on the left-hand side. Projects like the Atlas are supported by grants and especially by donations, so please consider giving a gift to the Oregon Bee Atlas at any level or contact us about endowments. Now, you can find a donation button on our website as well. Now, when you leave, you'll be prompted to complete a short survey asking you about topics you'd like to hear about in the future and the quality of the presentation. So please do take a minute to let us know your thoughts, because it will help us improve these lectures. Now, thanks for tuning in, and we'll see you in July with Olivia. And good night.